is the second part of sexual reproduction in flowering plants. So the second topic is gynoecium and megasporogenesis. All carpel in a flower consist of gynoecium. Flower can show monocarpellary or multicarpellary condition. And first, one nucleus cell from micropylar and become megaspore mother cell, which is MMC, which is diploid. It is further divided into four linear tetrad of which is the haploid megaspore. It further divided in four chelacell megaspore functional and three will be degenerated. So additionally, so it is further divided one functional megaspore, then divided, then cytoplasm of nucleus. It is divided first nuclear division, second nuclear division, which is second nucleated stage, then four nucleated stage, which is the second nuclear division, and third nuclear division, which is 8 nucleated stage. So again it is divided. Now the cell wall will be laid around these nuclei and matures embryo sac will be formed. I am feeling sleepy because of right now it is 1 a.m. So just chelacell end. So 3 antipodal cell in the top of the portion, 3 antipodal cell, then larger central cell, then 2 polar nuclei, then egg cell, which is the main female gamete, and second synergist, which is egg apparatus. Together egg cell and 2 synergists called the egg apparatus, and down filiform apparatus, finger like projection that guide entry of pollen towards the egg. So now it is the topic of pollination. There are 3 types of pollination: autogamy, geitonogamy and genogamy. Autogamy, which is self-pollination; geitonogamy, which is the transfer from anther to stigma of flower in a same flower. Different flower but same plant. And genogamy, which is the completely different from autogamy and geitonogamy. Genogamy pollination is called about the different plant and different flower. So viola, common pansy, oxalis and commelina produce 2 types of flower: chesmogamous and clistogamous, which is clistogamous flower closed, do not open. Invariably autogamous, clistogamous flower invariably autogamous. Biotic pollinators, majority and specific, for example moth and yucca. So floral reward. So agents of pollination, there are required agents of pollination. There are 2 types of agents: abiotic and biotic. Abiotic, which is non-living, called water and wine, and biotic, which is by insect, living actually. Biotic agents are living. So by insect, entomophily; by bird, which is called pollinator ornithophily; and by bats called chiropterophilia, by primates and reptile, etc. um, by wind, which is called anemophily. There are two type of abiotic agents, which is by bind or by wind and by water. By wind is called anemophily and by water is called hydrophily. Use of pollinator, which is amongst the biotic, then wind and water. So bear are widely used. Also, if you need my handwritten short note, then comment below, I will provide it. Also, you can watch this lecture 1.5x to for revision purpose only. Anemophily flowers usually have single ovule in each ovary and numerous flowers are picked in inflorescence. Here flowers produce large number of light and non-sticky pollen to be carried by wind. Stamens are also well exposed for very dispersal of pollen. Stigma is large and feathery to catch pollen. These cells of corn cab are actually the stigma and style that wave in vine to help the pollen. For example, vine pollination is very common in grasses. Hydrophily, it is quite rare and is shown by around 30 genera, mostly monocot, example Vallisneria and Hydrilla. Freshwater Juicera marine seagrass, example Vallisneria. Surface pollinations, female flowers use long stalk to reach water surface, and male flower also release pollen or surface. In sea grasses like Juicera ribbon-like pollens are released in water which reaches to female flower which is submerged in water. We have to note that wind and water pollination plants do not produce nectar and are not colorful. It does not require any colors. Pollen pistil interaction. Pistil has the ability to recognize pollen. Later it accept or reject it. Note, plant breeders understand and manipulate pollen pistil interaction to get the desired result. Artificial hybridization, it include emasculation and bagging techniques. So emasculation means eliminator masculiner, which is a male part, and bagging means stigma covered with butter paper. Anther removed from bisexual flower. Female male flower emasculated, then bagging, then butter paper is covered, and then bag remove and desired pollen dust, then rebagging. Bagging prevent contamination of unwanted pollen. Fruits and seeds will be formed after the bagging. Outbreeding, now the topic is outbreeding devices. There are 4 type of outbreeding devices. First, outbreeding device avoids synchronization. It is the features of either male part is mature before female part called protandry, or female part is mature before male part called protogyne. Anther gamete from different flower is needed, only autogamy is prevented, but geitonogamy can be occurred. Arrangement of anther and stigma on different position. Thus, pollen from same flower can't reach the stigma of a same flower. Here,